Staphylococci and related species. So just starting off with a patient scenario that you can reflect on as we go throughout the lecture. So the patient, 15-year-old female high school student tripped over a barrier and heard a pop in her right kneecap. Pain in the right knee and shin, unable to walk on the right leg. Patient was seen at an emergency clinic where it was determined that she may have torn her anterior cruciate ligament, ACL, and was referred to an orthopedic surgeon. Damaged ACL was removed and replaced with an ACL graft secured in place with screws. Patient was sent home with a brace to wear and a broad-spectrum antibiotic therapy. Follow-up appointment with primary care physician within a few weeks. Increased knee pain in the injured knee. Clear exudate noted at the incision site. Incision site was drained. Primary cultures and antimicrobial susceptibility tests were performed on a sample of the drainage at a large metropolitan laboratory. Many gram-positive cocci in pairs and clusters. Few polymorphonuclear neutrophils. So culture after 18 hours of incubation, heavy growth of coagulase-positive Staphylococcus species. A matrix-assisted laser desorption ionization time of flight MALDI-TOF mass spectrometry was performed on the isolate. The isolate was not identified as Staphylococcus aureus. Staphylococcus species based on the MALDI-TOF identification. Susceptibility test results use the latest reference ranges for minimum inhibitory concentration. The patient and her sister were frequent volunteers at a local animal shelter. The isolate was sent to the State Department for confirmatory identification, and it came back as Staphylococcus pseudo-intermedius. Pseudo-intermedius is specific to animals, but humans can become infected occasionally. So after reading this patient's case history, consider risk factors associated with acquiring this organism. So obviously the surgery was a major risk factor, but what about the surgery made it a risk factor? It wasn't just the fact that we opened up the body, it's also that there's a lot of tissue repair that has to happen. And the immune system can sometimes focus on the inflammatory responses instead of fighting the bacterial infection, so it kind of confounds healing at that point. The methods that were used to identify this isolate: coagulase methods for staph, and their limitations, the role of mass spectrometry can have in identifying common and uncommon species, and the importance of correct identification and using the latest susceptibility guidelines. So gram-positive cocci are common isolates in the clinical microbiology lab. Most GPCs are members of indigenous microbiota. Indigenous means it's found already inside of the body somewhere. Some species are causative agents of serious infectious disease. So the catalase reaction and appearance. Staphylococci are catalase positive. This is probably one of the most important distinguishing features, and this is a quick spot test that we performed to verify that it's a staph isolate when we're doing benchtop evaluation or workup of plates. If you recall from previous classes, the catalase test involves putting bacteria in the presence of hydrogen peroxide. If there's bacteria and hydrogen peroxide and you see bubbles, that means that it's catalase positive because it has that catalase enzyme that will break apart the hydrogen peroxide and produce gas from that reaction. On stain smears, it appears singly, in pairs, and in clusters, often referred to a bunches of grapes. Colony morphology appear creamy, white, or rarely light gold. Buttery looking, some species produce beta hemolysis. So for example, Staph aureus would be beta hemolytic on a sheep blood agar plate. They are non-motile, non-spore forming, non-encapsulated. Aerobic or facultatively anaerobic. One exception is S. saccharolyticus, which is an obligate anaerobe. Rare strains, small colony variants, 1/10 the size, and some of those are beta hemolytic. So Micrococcus resembles Staphylococci. Micrococcus luteus is a pretty common microorganism that we see. It is a GPC, catalase positive, coagulase negative. It has a distinct yellow pigment. So on a blood agar plate, you're going to see this yellow pigment color. That yellow color is really evident if we do grow it on like just a nutrient agar plate. It's generally considered a non-pathogen unless the person is significantly immunocompromised and it's found in certain parts of the body. So in general, we don't worry too much about this organism causing infection in normal, healthy individuals. So differentiating Staphylococci and micrococci. So one of the major things for the qualifying exam and for this course is being able to create flowcharts that will help you keep different species separated in your mind. So it's a portion of the assignments, and then having those in the back of your mind will also help you answer those questions when you get to taking tests for the course or taking the test questions on that qualifying exam. So these tables are good for you to reference. I recommend making a physical flowchart where you start with Staphylococci and micrococci, and then have two little arrows like going out and saying this group of staph is coagulase positive, this group is coagulase negative, and then you can kind of take it further and further and further from that. So the coagulase test. We classify Staphylococci as coagulase positive or coagulase negative. The staphylocoagulase enzyme clots plasma. Coagulase positive staph are able to produce this enzyme and they clot the plasma. Staph aureus is the predominant coagulase positive human pathogen, so Staphylococcus aureus. S. delphini, intermedius, lutrae, hyceus are animal pathogens. You don't necessarily need to know all of the animal pathogens. Most of your qualifying exam is going to focus on human pathogens. However, when I took the test, I did see some species on there that were not familiar to me, so they were likely the animal pathogens that were on there. Coagulase negative Staphylococci, CONS. This is the abbreviation, and it's very common to see that even when we report out the identity of certain test results, you'll see it's just reported as coagulase negative. S. epi can cause hospital-acquired infections, but Staphylococcus epidermidis is also just found on the surface of your skin everywhere. So that's probably the most common skin bacterium that you're going to find. If a blood culture tube turns up positive for Staph epi, we usually want to do like a recollect to make sure it wasn't just contamination from the patient's own skin that caused that to turn positive. It is a commonly known natural normal flora of the skin, so that's pretty common to find that. But it can cause hospital-acquired infections. Staphylococcus saprophyticus can cause urinary tract infections in young and sexually active females. It's important if we find that in urine samples. Clinical significance of Staph aureus. This is the most clinically significant Staphylococci. It causes various cutaneous meaning the skin, infections and purulent abscesses. Purulent means it has a lot of pus, neutrophils, abscesses. Common cause of infective endocarditis. Endocarditis is an infection of the heart valves within the heart or the inner tissues of the heart chambers. And then there's toxin-induced diseases, food poisoning, scalded skin syndrome, and toxic shock syndrome. Coagulase negative staph, most significant species are listed here. S. lugdunensis versus Staph aureus, both are slide coagulase positive, but the tube coagulase test is crucial in differentiating them. Virulence factors of Staph aureus, so there are enterotoxins, cytolytic toxins, cellular components such as protein A, exfoliate toxins, and despite these virulence factors, innate resistance to Staph aureus is high, and the organism is regarded as an opportunistic pathogen. So even if a person tests positive for carrying Staph aureus, we usually assume that they are asymptomatic carriers and it's not going to cause a problem unless their immune system is weakened or something else is going on inside of their body. So enterotoxins, these are heat-stable exotoxins that cause diarrhea and vomiting. There's toxins A through E and G through J. There's 9 total. They are resistant to gastric acid. A, B, and D are associated with food poisoning. For this course, you do not need to memorize all of the toxins that are produced by all of the species of microorganisms that we're going to talk about. The main thing to know about these toxins is that it can cause the diarrhea and vomiting and food poisoning. So what happens when you make a turkey, for example? A lot of times you'll read the instructions for how to thaw a turkey, and it'll say thaw in a refrigerator. So then if you don't thaw a turkey in the refrigerator, if you just put it on the counter overnight and you wake up the next morning and you feel that that turkey is now room temperature, and we don't know when it turned room temperature, maybe it turned room temperature and was completely thawed in the middle of the night. The longer that turkey is allowed to sit at room temperature, the more that Staph aureus bacteria can grow on the surface of the turkey. And it's not going to be on every turkey. We do a lot of food processing and trying to clean off all the bacteria from the turkey, but it's possible that there could be some Staph aureus on the turkey. And if it's at room temperature, that's a pretty good temperature for it to start to make copies of itself. While it's copying itself, it is releasing these exotoxins, and the exotoxins are being deposited on the surface of the turkey. You take that turkey, you put it in the oven for however many hours, you cook it to the perfect temperature, everything's cooked and all the bacteria is killed, but those toxins are heat stable, so they do not deactivate in the presence of the heat in the oven. So then when you consume that turkey, you are consuming toxin, and that toxin then gets in your digestive tract and causes self-limited diarrhea. So it can stay in the body for a brief period of time, but usually between like 8 and 24 hours, that toxin is completely flushed out of the system and you are cured from this limited food poisoning. The reason why you don't remain sick for longer is because the bacteria itself was killed in the oven. It's just the toxin that was left behind. And toxin isn't a living organism, it's protein. So it doesn't like replicate and make copies of itself. Once the toxin's flushed out of the body, it's gone. Toxic shock syndrome toxin is chromosomal mediated toxin. TSST1, B, C, G, and I are superantigens. It causes toxic shock syndrome. Then there's an exfoliative toxin. There's 2 types, A and B. It causes the epidermal layer of skin to slough off, and it's known to cause scalded skin syndrome or Ritter's disease. Also implicated in bullous impetigo. Cytolytic toxins, extracellular proteins that affect red blood cells and leukocytes. Hemolysins and leukocidins. Staph aureus produces 4 hemolysins, alpha, beta, gamma, and delta. Alpha hemolysin lyses red blood cells and damages platelets and tissues. Beta hemolysins shows enhanced activity by acting on the sphingomyelin of red blood cell membranes, causing lysis. And then there's a hot-cold lysin because it works best at 37 degrees Celsius and very well when stored at 4 degrees Celsius. Gamma hemolysin, delta hemolysin, these are flip-flopped. I don't know why those are flip-flopped there. So this one is actually delta and this one is actually gamma. But either way, delta hemolysin causes injury to cells and leukocytes, but it's less lethal. Gamma hemolysin, also called Pantin-Valentine leukocidin. Exotoxin kills polymorphonuclear leukocytes and helps suppress phagocytes, often associated with community-acquired infections. Enzymes. So the exact role of enzymes is uncertain, but it's considered as a virulence marker. Coagulase is very diagnostic, but importance in virulence is not completely understood. So we leverage that coagulase enzyme in order to diagnose or confirm diagnosis of certain staph species, but it's really unknown why it exists. Hyaluronidase hydrolyzes hyaluronic acid present in connective tissues, helping the spread of the infection. Lipase breaks down fats and oil created by the sebaceous glands on skin surfaces. That allows it to stay on the skin surface and consume nutrients from that surface. Protein A. So there are some diagnostic kits that we use for staph, and one of them is a latex bead assay. And that latex bead assay has latex beads that have antibodies that are designed to bind to protein A and coagulase. And when it binds to both of those, it will form clumping, and then you can visibly see that in the clumping. But protein A, as a virulence factor, it binds to the fragment crystallizable Fc portion of antibodies to avoid phagocytosis, masking of its immunogenic properties with host proteins to look like self. It assists in blocking phagocytosis, and it negates the protective effects of IgG. So Staph aureus, the primary reservoir, is in the nose or in the nares. So when you get tested for Staph aureus to see if you're a carrier, they do a nose swab. Other reservoirs would be the axilla, vagina, pharynx, and other skin surfaces. It occurs in hospital outbreaks, nurseries, burn units, surgical patients, and transmission is primarily by unwashed hands and inanimate objects known as fomites. This really harps on the importance of washing hands when going from one patient room to the next or touching one person to the next, and even working in the hospital laboratory. Mode of transmission, traumatic introduction. So needle stick, destruction of skin layers, burns, road rash, medical procedures, cuts, anything like that. Predisposing conditions, so any type of chronic infection, indwelling devices, skin injuries, or immune response deficits could cause infection with Staph aureus. Skin and wound infections are opportunistic that can be the result of previous skin injuries. They're pus formers, can form a furuncle or a boil, and that's a painful inflammation of the skin and subcutaneous tissue. Or carbuncles, boils that have multiple lesions and may progress into deeper tissues. Folliculitis is mild inflammation of a hair follicle or oil gland. Bullous impetigo, large pustules surrounded by a small zone of erythema or redness, spread by direct contact, and fomites, and that's highly contagious. Sometimes secondaries to skin diseases occur due to blocked follicles, sebaceous glands, and sweat glands. Scalded skin syndrome, a bullous exfoliative dermatitis. It occurs primarily in newborns and previously healthy young children. It may occur in adults, but more likely to occur in renal failure patients and immunocompromised patients. The severity of the disease ranges from localized skin lesions in the form of a few blisters, pemphigus neonatorium, to a more extensive condition. It affects 90% of the body, or if it affects 90% of the body, it's called Ritter disease. Spontaneous recovery incidence in children is high. So basically what this thing does is it releases that exfoliative toxin, and the toxin goes to the surface of the skin and causes the skin to slough off, and it sloughs off in large pieces. And it is very common in children, less common in adults. And if an adult gets it, it's usually really detrimental and can be fatal for an adult. Children usually have a high incidence of recovery with no prolonged complications. Toxic shock syndrome is a rare multi-system disease associated with superabsorbent tampons. Its clinical presentation is the sudden onset of fever, chills, vomiting, diarrhea, muscle aches, and rash. Extreme cases lead to hypotension, which is low blood pressure, and shock. It's fatal in 2 to 5% of cases due to the multi-organ system failure. The food poisoning I already talked about, those enterotoxins, usually the food handler, or it's already on the surface of meat that needs to be thawed out. It's on contaminated food. So modes of transmission: improper food handling and storage, or food stored in room temperature. Commonly contaminated foods include salads, meats, poultry, egg products, select bakery products with cream fillings, sandwich fillings, and dairy products. There's no obvious signs of contamination when it's present. Symptoms appear rapidly about 2 to 8 hours after ingesting, usually resolve in 24 to 48 hours, sometimes less. Nausea, vomiting, abdominal pain, cramping, diarrhea, and headache is also possible. Death is rare but may occur among older patients, infants, or severely debilitated people. Other infections caused by Staph aureus: bacteremia and endocarditis. So IV drug addicts presenting with fever enter through the injection site. Secondary pneumonia: after influenza virus infection, it has a high mortality rate. Osteomyelitis occurs secondary to bacteremia and results in bacteria invading in the bone. Fever, chills, swelling, and pain around the infected bone area. And then septic arthritis. S. epidermidis, as I mentioned, is usually considered normal skin bacteria. It's a common source of healthcare-acquired infections, though. Predisposing factors: skin flora gets introduced into catheters, heart valves, cerebrospinal fluid shunts. Organisms often produce a slime layer or biofilm that helps adherence to prosthetics and avoidance of phagocytosis. S. saprophyticus. So S. saprophyticus can cause UTIs in young sexually active women. It's due in part to increased adherence to epithelial cell lining in the urogenital tract, and it's the second most common cause of uncomplicated cystitis. It's rarely present in other skin areas or mucous membranes. In urine cultures, it is considered significant even when it's present in low amounts. Staphylococcus lugdunensis can cause both community-acquired and hospital-acquired infections, can be more virulent than Staph aureus, and can clinically mimic Staph aureus infections. It is known to carry the MEK-A genes. That is the gene that encodes oxacillin resistance, also known as methicillin resistant. So if you've heard of MRSA, methicillin resistant Staphylococcus aureus, this one is also known to carry that gene. It's an important pathogen in infective endocarditis, septicemia, meningitis, skin and soft tissue infections, UTIs, and septic shock. Aggressive in endocarditis, causing valve replacement, and infections have a high mortality rate. Other coagulase negative, so just as a review, Staphylococcus aureus is coagulase positive. Staph epi is coagulase negative, so other coagulase negative organisms. There's opportunistic pathogens that are listed here. Normal flora, but can be involved in endocarditis, septicemia, and wound infections. Staph hemolyticus is a common coagulase negative staph, can be found in wounds, bacteremia, endocarditis, and UTIs. It may have resistance to vancomycin. Staph pseudointermedius is a common cause of pyoderma in dogs and skin, ear, and post-operative infections in dogs and cats. It has been linked to human infection. Infections are known to occur in humans at the surgical site: rhinosinusitis, catheter-associated bacteremia. Veterinary staff and pet owners are at risk of zoonotic transmission. Zoonotic transmission is talking about the transmission between different species. It's known to be resistant to oxacillin and other antibiotics. So this one's kind of like the MRSA for dogs. So working up specimens for staph. If we suspect a staph infection, there's no special procedures that have to be followed. You just take the specimen from the site of infection, cleanse the surrounding area to avoid contamination with like that staph epi that's all over the surface of the skin. Transport to the lab without delay. It prevents drying and maintains the proper environment. It also minimizes the growth of contaminating organisms. And then you look at it under the microscope, you can expect to see many gram-positive cocci, possibly some polymorphonuclear leukocytes. So in this picture right here, all of these pink cells are like neutrophils. And that's what makes up the bulk of pus. And then these are the cells in clusters. That's your gram-positive cocci in clusters, and that's when you would suspect a staph infection. Purulent exudates, joint fluids, aspirated secretions. Aspirate is best. If you're using a swab, clinicians should send 2 swabs. 1 swab is usually for the gram stain, the second swab is usually to work it up on the plates. Aspirated abscesses showing GPCs and PMNs over here. Here's microscopic morphology of staph, so you can see the clustering of the gram-positive cocci. Isolation and identification of staph. It grows easily on a sheeplet agar plate and in thioglycolate. If it's heavily contaminated, you can use one or more of the following selective media. Mannitol salt agar, MSA. So previously I had talked about what MSA is. It has the mannitol sugar. Staph aureus is one of the organisms that would turn yellow on this, but there are others that can utilize the mannitol sugar. CNA, PEA, those will basically try to get rid of the gram-negative organisms that we're not interested in growing. And then there's chrome agar for Staph aureus. It is a selective media. Chrome is referring to color changing properties. So there's like MRSA chrome agar, and there's different types of chrome agar that exist that will turn different colors if it has particular features. So cultural characteristics: round, smooth, white, creamy colonies on SBA after 18 to 24 hours. Staph aureus can produce beta hemolytic zones around the colonies. It rarely exhibits a yellow pigment when extended incubation. SCVs, non-pigmented, non-hemolytic pinpoint colonies. That's those small colony variants. So this is a picture of Staph aureus growing on an SBA plate. So pretty strong, robust colonies. They're creamy, and you can see that zone of beta hemolysis around it. Staph epidermidis colonies are small to medium-sized, non-hemolytic, and they're gray to white. Some strains may be weakly hemolytic. Staph saprophyticus produces slightly larger colonies, with about 50% producing a yellow pigment. Hemolyticus grows as a medium-sized colony with weak or moderate hemolysis and variable pigment production. Lugdunensis colonies are often hemolytic and medium-sized. The identification of Staphylococci should not be done on the basis of colony morphology alone. As you start to look at more and more plates and you start to receive plates from patients, you kind of get a feel for what organism you think it is based on what it looks like on the plate. But you should definitely always follow it up with confirmatory identification of 2 or 3 additional tests besides just colony morphology. Here's coagulase negative staph on an SBA plate. So other types of identification methods, the traditional method would be OF glucose medium. Most staph ferment glucose, micrococci do not. OF testing doesn't work well for all staph. So we do other types of tests, like there's a modified oxidase test that's available. And then there's multi-test systems that are incorporated into the traditional biochemical tests. Otherwise, we can do molecular testing for group B. Sorry, we'll save that for the next lecture. So for molecular testing, we can test for the presence of like the MEK gene if we're looking at Staphylococcus aureus to see if it is a MRSA. Plasmid typing or MALDI-TOF mass spectrometry. The MALDI is one of the go-to tools for diagnosing the various species of staph. So again, this is a good chart that can help you differentiate amongst the different staphs. It's a good thing to take a look at. You will not have to know all of these species for this course or for the qualifying exam, but just kind of taking a look at what tests are performed to differentiate gram-positive cocci. The tube coagulase method detects staphylocoagulase or free coagulase. Staphylococci is extracellular molecule that causes a clot to form when bacterial cells are incubated with the plasma. It reacts with a thermostable thrombin-like molecule called coagulase reacting factor to form a coagulase CRF complex. This complex resembles thrombin and indirectly converts fibrinogen to a fibrin clot. The clot formed in the tube may undergo autolysis. Look for clot formation after 4 hours of incubation. If no clot appears, the tube should be left at room temperature and checked the next day. It's important to note that other species other than Staph aureus produce a clumping factor. So this is the tube coagulase test, and you can see the clot that has been formed here. And here you can see the coagulase positive Staph species. The PYR test differentiates Staph aureus from other Staph species. This test, the substrate, is hydrolyzed and to a couple different substances, which combines with I'm not even going to try to pronounce that word, dimethylaminocinnamaldehyde to form a red compound. So red color change would be a PYR positive test. I haven't really commonly seen this done on Staph species, more often strep species, so we're going to circle back to this test once we get to talking about strep. So if it's a urine sample, we want to test for Staph saprophyticus. A presumptive ID can be done using novobiocin. So we streak the organism onto the plate, add a novobiocin disc to a heavy growth quadrant. If it's resistant, we can assume it's Staph saprophyticus. If it's susceptible, it's likely Staph epidermidis. So this is the novobiocin susceptibility test. So here you can see this is susceptible, this is resistant. Going back to here, if it's susceptible, it's likely Staph epidermidis, so Staph epidermidis down here. If it's resistant, Staph saprophyticus, so Staph saprophyticus would be resistant. This is more of like the schematic chart that you should be doing in your assignments for the course. So when you see gram-positive cocci, you would perform the catalase test and kind of go from there with the differentiation. I've seen many different versions of this chart. It's important that you create your own version and you study your own version, because that's what you're going to remember and internalize when it comes time to take that qualifying exam and even to take the tests for this course. Rapid ID, I did talk a little bit about the agglutination kits. So there's several agglutination kits that exist for differentiating Staph aureus from coagulase negative staph. And this is a picture of one of those kits. I talked about the latex beads that will detect clumping factor or coagulase and protein A. We always follow up the rapid methods with culture and a MALDI or some type of DNA test. Third generation agglutination kits detect protein A clumping factor and antibodies that bind capsular antigens 5 and 8 or other surface molecules. So those are more sensitive but less specific. False positives can occur, so careful consideration of source colony morphology and susceptibility pattern can eliminate some of those errors. PCR can be used, especially if we're trying to detect that MEK-A gene to determine if it's MRSA. Molecular methods are used directly to identify staph from a positive blood culture. So now we have a blood culture system where we have the most common organisms that would be found in a positive blood culture, and we have the a multiplex type PCR test that can be performed on blood sample directly. So if that organism is present in the blood, its DNA would be present in the blood, and we'll be able to evaluate that DNA sample and determine which species it is. If we do see Staph epi in a blood culture that turned positive after like 5+ days of incubation, we kind of assume that that was a contaminant. Newer technologies are being evaluated. Even not too long ago, I was in a conference and they were talking about multiplexed type molecular analysis for site-specific areas. So they'd take a sample from a particular site and use a kit that's specific to that site to evaluate the different organisms that could be found. MALDI, again, continues to gain momentum. It is building its database of all the different organisms that it can accurately identify, and every time they come out with a new list of organisms that it can successfully identify. So characteristics use standard guidelines issued by CLSI. Most Staph aureus isolates are resistant to penicillin. Beta-lactamase testing should be done. Serious infections do require susceptibility testing. Methicillin-resistant Staphylococci. So there's MRSA, there's MRSI, there's CA MRSA, community-acquired, healthcare-associated MRSA, and hospital-associated MRSA. Control of MRSA infections, barrier protection. So you want to make sure that you are protecting yourself when going into a patient's room. Contact isolation, hand washing. The treatment of choice for MRSA is vancomycin, but concerns remain because of the resistance to glycopeptides, call for restrictive use of these drugs, and selective laboratory reporting. Vancomycin also has some significant side effects, so a lot of patients get sick from taking vancomycin. Laboratory scientists must always refer to the latest susceptibility guidelines for correct antimicrobial agent interpretations. So MRSA, heterogeneous resistance to beta-lactams, does occur. One subpopulation is susceptible, another subpopulation is resistant. The growth of the resistant subpopulation is enhanced at neutral pH, sodium chloride concentration of 2 to 4%, lower incubation temperature, or prolonged incubation. Media containing oxacillin, or preferably cefazidin, can be used to screen for MRSA in clinical specimens. These media can also differentiate MRSA isolates from isolates that are hyperproducers of beta-lactamase or borderline oxacillin-resistant Staph aureus that do not grow on those plates. Chromogenic media can be used to identify MRSA directly from clinical samples. So basically with this one, you take a nasal swab, smear it on the plate, and rub it for like isolation on a plate. And if it is positive, it'll change a particular color. So most of them that I've seen turn blue. I have seen some turn pink in certain kits, depending on what type of chromogenic media you're using. So you just want to go with the manufacturer's instructions of what you're seeing. There are automated antimicrobial susceptibility systems using cefazidin, generally available to identify MRSA. Most oxacillin resistance is caused by the MEK-A gene. It's carried on mobile cassette known as the SCC-MEK. This gene codes for an altered penicillin binding protein that does not bind to oxacillin, rendering the drug ineffective. Most oxacillin resistance is caused by that MEK-A gene. Latex agglutination tests are available to detect altered penicillin binding proteins. The gold standard, though, for that detection is nucleic acid probes or PCR amplification. Vancomycin-resistant Staphylococcus aureus, vanco-resistant S. aureus, VRSA, and vancomycin intermediate Staph aureus. Often in patients with underlying conditions, it's important to adhere to infectious control practices and guidelines to limit the emergence of this highly resistant organism. So if it is resistant to vancomycin, that's kind of our last resort drug. So that definitely has to be reported, and that becomes an emergency situation. Laboratories should consider using more than one method to detect VISA. It's important to confirm VRSA by a reference method and adhere to the CDC guidelines. Macrolide resistance. So resistance to the macrolide clindamycin may not be obvious. Erythromycin and clindamycin normally have the same resistance patterns, but if the susceptibility patterns of these two antimicrobials are different by standard testing methods, additional testing uses a D-zone test that might be useful. So staph resistance to clindamycin is occasionally inducible, meaning that it's detectable only when the bacteria are also exposed to erythromycin. Inducible resistance in this case can be detected by the D-zone test. So you place an erythromycin disc near a clindamycin disc and use the latest performance standards for susceptibility testing. If the isolate possesses inducible clindamycin resistance, there's flattening over the clindamycin zone between the two discs, giving a D-shaped zone of inhibition. Some automated systems have the ability to detect this resistance. So this is the erythromycin, the clindamycin disc, and you can see the flattening. So this is inducible resistance. So over here, it's super susceptible, but when you put it near erythromycin, this resistance can be induced.